Thank you for joining us today. My name is Yannick Goriab, and I'm the Health Information Specialist with Brain Tumor Foundation of Canada. We are very excited to offer another webinar series for 2014. This year we will have 12 webinars, one for every month. Some of the webinars will be designed for healthcare professionals and others will be for a wider audience, including helpful information for those affected by a brain tumor. We will have all of our webinars listed on our website very, very soon. And today marks our very first of 12 webinars, and we have Dr. David Stroudel joining us from Ottawa, Ontario. Dr. David Stroudel is a scientist at the Children's Hospital of Eastern Ontario in Ottawa, Canada, with appointments in the Department of Pediatrics and Department of Biochemistry, Microbiology, and Immunology at the University of Ottawa and the Ottawa Hospital Research Institute. Dr. Stroudel group has focused on understanding host-virus interactions critical to oncolytic therapy as a guide to developing more effective virus platforms. This has led to the development of several clinical candidate viruses, including VSV-M51 and Maribut MG1, used by member labs throughout the COVCO program. As a complement to these studies, Dr. Stroudel group has developed a high-throughput functional genomics program to define host genes that modulate oncolytic therapy in an effort to define opportunities for optimally effective combination treatments. These screens are the first reported application of genome-wide RNAi screening to a biotherapeutic and have recently been published in Cancer Cell. Dr. Stroudel is the director and developer of the CHIO Automated Screening Center and the CHIO IDIS Imaging Facility, and is a scientific co-founder of Generex Biotherapeutics in San Francisco, USA. Dr. Stroudel presentation is titled Oncolytic Viruses in Clinical Trials, New Horizons of Brain Cancer Therapy. Welcome, Dr. Stroudel, and I'll hand it over to you right now.
Thank you very much. I hope everybody can hear me. It's sort of an isolating phenomenon here, not being able to see the audience. But I would like to thank the Foundation, Brain Tumor Foundation of Canada, for the opportunity in this really unique information session, this webinar session, to allow us to talk a little bit about our perspectives on what the new horizons might be in terms of treating a very difficult clinical challenge, which is that of cancers in the brain. And so it's really a presentation that is not going to be data heavy. It'll be more in terms of introducing some of the concepts and some of the challenges that we're attempting to deal with in trying to treat brain cancers. So, without further ado, we'll talk a little bit about brain cancer facts. And so, brain cancer is roughly divided into two types of cancers. Although this is really a historical designation, cancer in the brain, as other cancers throughout the body, we're starting to realize are in fact not diseases that are defined by the location in the body, but more the genetics of the individual cancer and such. And the reason that brain cancer is really not just two diseases, primary and secondary, is because there's hundreds of different kinds of brain tumors, and they're all defined really by the genetic signatures that underpin those and give rise to the driving mutations that make those cancers what they are. But primary cancers basically are operational definition in that they arise in the brain, and about 2,500 patients in Canada present to the clinic with a new brain tumor each year. And that scales roughly globally with population. So for example, in the US it would be perhaps 10 times that many again each year. So it's a significant problem. Now 50% of those brain tumors, those primary brain tumors, are actually gliomas. And of those gliomas, 50% are of the very aggressive form, glioblastoma multiforme. So it's called GBM for short, and I'll probably use that acronym on and off. And the problem with this advanced glioma is that the prognosis are very challenging, and in fact our attempts to do anything about this disease is largely unchanged over the last 30 years. Although there's been many incremental improvements, there's not been many great changes to the overall survival and the condition of these patients. So it really is an unmet clinical challenge. And the other kind of brain cancer is so-called secondary brain cancers, or brain cancers that metastasize from other tumors in the body into the brain. It's much more prevalent, so there's many more times these patients presenting in the clinic. And it's a different disease, but we feel that many of the same challenges that we see with primary brain cancers are also the same for metastases to the brain, and I'll speak about some of those challenges to effective therapies. And so consequently, when we're thinking about treating brain cancers, we often are thinking about trying to deal with both of these classes of tumors, brain tumors. I'll also mention that brain cancer does not discriminate by age. In fact, it's the leading solid tumor, killer of children that have solid tumors. And so we are also focusing our attention, as are many researchers, in trying to deal with this, not just in adults, but in children as well. Currently, the way we treat many cancers, and that's the same for brain cancer, is to use surgery as well as radiation and chemotherapy. Surgery is to remove the tumor mass as best as possible, and then we use radiation therapy, both focal and field radiation therapy, to try and get at the residual cancer that was not removed through surgery, and then chemotherapeutics, either concurrently or after, to try and get at those cells that we couldn't find, the surgeon couldn't resect, or that have gotten away from the original tumor. And that, although it is our standard of care, it's state of the art, too often it's just not effective enough, and that's why, of course, we're speaking about this today. So why is brain cancer so difficult to treat? So the first thing is that, at least for gliomas, and it's true for other brain cancers, they're highly invasive tumors. So here's just a picture, this is a mouse model cancer, but very similar in the patient. If you could fluorescently tag your tumor, and if it was of the type that we see in this picture, where it remains a very tight border around this tumor, so this green area is tumor cells, and the black area here are normal cells in which this tumor sits. And there's a very discrete border between them. If we could find this tumor and define that border, and the surgeon could just sort of cut along the line here and remove that tumor, then perhaps this tumor would not reoccur. And that's true in other cancers, and why surgery is still the most effective cancer therapy that we have in our arsenal. However, gliomas, many brain cancers, are not like that. They don't form those discrete borders. In fact, you have cells that are highly motile, and they move away from the original tumor and spread into the parenchyma, the rest of the healthy tissue in the brain, and often at great distance. So here's just a sort of 30,000-foot view of this tumor. And you can see the primary tumor here, and you can it's stained in brown. And you can see that other tumor cells have sort of left and set up other tumor elsewhere. And that can be locally, but it can also be all the way across to the other side of the brain before these patients even present with an issue in the clinic. So you can see why surgery is simply not a curative modality for this therapy, because for this disease, because this horse essentially left the barn before the patients can be seen by a doctor. So that's a great challenge. The other great challenge is that these tumors reside in incredibly complex and delicate tissue. So this is just sort of a cross-section of the brain, and in yellow we can see neurons that have been stained. And you can see they travel over great distance, and they interact in these very complex networks. And in fact, this is critical for how the brain functions. It's a very intricate network of cells that communicate with each other at high speed and great distance. And consequently, you can't remove much of this tissue. Oops. You can't remove, if a tumor is residing inside here, you can't really rip out all this tissue and hope that it can regenerate itself, as is the case in many of the other organs outside of the brain. So it's a very difficult tissue in which to have a tumor reside because of the delicate and complex nature of the tissue. So consequently, what we need here are therapies that are highly selective so that we don't have to damage this very intricate structure and lead to the debilitating side effects that occur. Another issue is the blood-brain barrier. Now, all the parts of our body are fed with blood and so on, and that's done through our circulatory system, the blood vessels flowing through our body. But in the brain, there's a special change in our blood system that has been called the blood-brain barrier, in that the cells that line, this is just sort of a cross-section of a blood vessel, the cells that make up these blood vessels are very, very tightly packed with these tight junctions that seal these cells together very, very tightly. So consequently, the things that are flowing through the bloodstream have a very difficult time getting out of the bloodstream into the surrounding area. So for example, if you had a tumor somewhere here and you delivered your drug through the bloodstream, it'd be difficult to get the drug out to that tumor because of this sort of very tight structure. And that's been a challenge for developing drugs. In the past, you can get a drug that perhaps works when you put it directly onto the tumor cells, but when you try to deliver it through the bloodstream, it simply can't get out, and so it's basically cuts down the number of drugs that we have in an effective arsenal against brain cancers. So those are sort of easy things to see, sort of obvious things about brain cancers that make it an extra difficult challenge. But there are now things that are slightly more subtle that's taken years of research and technological advances to be able to understand. And sort of the first part is depicted here on the left, in that it turns out the genetics of a particular cancer is quite different between patients. So the so-called inter-tumor heterogeneity. So if 10 patients present to the clinic with similar tumor, a lung tumor, and then you were to sequence those, find out the genetic makeup of those tumor cells, those tumors would be very different across this population of 10 people. And so consequently, if you design a drug that works in this particular tumor because of the genetic deficiencies in that tumor, it might not necessarily work for these other people because, in fact, they have a different disease because the genetic underpinnings, the deficiencies in the genetic makeup of those tumors that have caused them to be malignant are different. Then it's very difficult to make one drug to fit all. And then you have a situation where drugs fail in the clinic because they're only effective in a very small population of people. So that's clearly a challenge that's happening not just for brain cancer and all cancers, but it's certainly true for brain cancer as well. And it's very difficult to figure out which drug to match with which patient because those tools and understanding are only now being figured out. And there's an very active study to try and personalize your drug therapy to your particular genetic disorder or genetically driven cancer. Now, if the story ended there, then it would actually still be quite hopeful. But the issue is, we've now realized that by sequencing a particular person's patient in more detail, a particular patient's cancer in more detail, and particularly situations where you have one cancer that's metastasized to a second location, and sequencing both of those, you find, in fact, that even within a patient, the genetic makeup of their cancer is also very different. So you have pockets of this type of genetic makeup mixed in with this type, mixed in with this type. And so now, even within one patient, you're actually having to treat more than one disease. So it's now, we now understand why drugs have failed in many cases, because we're actually trying to treat multiple diseases with a drug that will probably only work on one part of this. And so then this part reoccurs and the cancer comes back. So it's a very difficult challenge to try to treat cancer with a single therapy or a therapy designed for a particular makeup of genetic deficiencies. Now, sort of along those lines, but slightly more intricate, is the fact that there are in fact different kinds of cells within a tumor, not just the genetic makeup, although the genetic makeup will define what kind of cell you are, but in fact cells that have sort of different jobs, so to speak, in the tumor. And this is a concept that was, it's a homegrown story, in fact, a Canadian story. This concept of the stem cell was born in Toronto in the 60s and has in fact stayed in Toronto. There's been some really landmark discoveries by Toronto scientist John Dick, who found a cancer stem cell, the first cancer stem cell, a leukemic cell. And later on, Peter Dirks, another Canadian scientist, clinician scientist, defined the cancer stem cell from brain tumors. And this is the idea that not all cells function, have the same role to play within a tumor, for example. If you liken this to a hive of bees, the queen bee could be thought of as this cancer stem cell. It's a cell that gives rise to more of itself and more of these drone cells that make up the bulk of the tumor. So you have drones and queen bees all tucked away inside this tumor mass. And the vast majority of them are these drones that make up the tumor mass, and they're susceptible, and we've been building our drugs to target these drones because those are the ones that we've worked with in the labs and so on. And they're targeted with our chemotherapies and our radiation, and we set our doses based on those and so on. And we can decrease the mass of this tumor with these drugs. But as it turns out, these cancer stem cells, or these queen bees, they're resistant to chemotherapy and radiotherapy. And so they'll remain after we've treated them. And because they're cells that are able to give rise to many, many more drones, they just repopulate the hive or repopulate the tumor, and the tumor relapses. And so when we now start thinking about how to permanently treat these tumors, we need to now fold into the concept that there are different kinds of cells that we need to be examining when we're testing our therapeutics to make sure that we're killing all of the cells, both the drones as well as these cancer stem cells that reside inside the tumor and repopulate the tumor. So what needs to be done so that we do better clinically? Well, many of these are obvious. First, we need to target cancer that we can't see, that we can't resect. It's especially true in brain cancer. Again, it's a very delicate organ. So how do we deal with this disseminated nature of the disease in an organ that we can't really root around in as we like? The second is we need to be targeting the appropriate cells within the tumor, these cancer stem cells. Now, probably not just the cancer stem cells. We need to kill those drone cells as well, but we need to certainly kill the root of these tumors, along with the large masses that are being dealt with. And we also need more potent therapies. We need therapies that inherently are just better at killing, but at the same time are much more highly selective for tumor cells, such that they, while we're busy adding potency, we're also not increasing the level of damage. Again, in an organ where we can tolerate very little damage. So how do we go about doing that? What are the concepts that are floating around now that might address some of these really challenging issues in the clinic? Well, the first thing is that we perhaps need to go back and rethink the way we've decided to find drugs to treat brain cancer. And that's because we've probably been targeting the wrong cell. Instead of targeting all these blue cells, which is appropriate, we need to target those. We also need drugs that are going to hit this cell here, the cell that's able to repopulate and replenish this whole tumor, this so-called cancer stem cell. And in the past, we haven't been building our drugs using this as the target. We've used these blue cells as the target and found drugs that are capable of being killed by those agents. But these have inherently different properties, the genes that drive these cells are different, the replication cycles are different, and so consequently their susceptibility to drugs will be different. And we need to go back and do what was done 20, 30 years ago to find new drugs that can be used to target these cells as well. And so some of the concepts that are being thrown around now is to find drugs that can kill those cells. But another interesting concept that I'd heard Peter Dirk speak about is this idea that, well, these queen bee cells, their job is perhaps to repopulate cells. If, in fact, they've taken over the genetic programming that our bodies use to repair damage in the cell. We have stem cells whose job it is to do that. These malignant forms of those stem cells, their job is to make more of themselves, and then those progenitor cells that they give rise to then go through this process called differentiation. There's a few genetic changes in those cells that cause them to be functional members of that tissue. They'll do their job as a brain cell, a neuron or an oligodendrocyte, whatever those cells are supposed to do in the tissue. But once they've formed that type of cell, they no longer can renew themselves anymore. And so an interesting concept might be to find drugs. If you can't find drugs to kill these cells, perhaps you can find them drugs that push them from this red cell into a blue cell, so that they go down a one-way street and they can't come backwards to be a rejuvenating cell. They instead will become a terminally differentiated cell that sits in the tissue and can no longer make more of themselves. And in that way, they essentially be there but be rendered harmless and not contribute towards the cancer anymore. So there's some interesting concepts that are being examined. It's still in the very early stages. But clearly the idea of trying to target these very important members of a tumor, a brain tumor, is a key concept that may bear fruit in the near future. Another interesting concept that looks very, very promising is this idea of immunotherapy. And this is the use of the repurposing or the retargeting of our immune system to target cancer cells. And this is a concept that is actually very, very old. It's decades old, the idea of taking our immune system and directing it against tumors. But up until very recently, there's been a lot of activity, but there's been very disappointing results. And most of that is because it's taken us until very recently to know enough about how the immune system works to be able to repurpose it effectively. And recent results in many different groups in the world have been so promising, in fact, that one of the most prestigious cancer journals, Science magazine, has just last month bestowed a breakthrough of the year to cancer therapy as a modality to treat cancer. And I'll explain a few reasons for why that was. So our immune system is made up of many different cells in the body and chemicals that form a very intricate network of communication. And its job, generally speaking, is to identify foreign invaders as well as cells that are not behaving properly in the body, so domestic invaders, and launch a response to eliminate those potential pathogens. And it's broken up into roughly early responders and late responders is one way to look at them. These cells here are cells that are able to survey the body and find irregular cells that have been infected with a virus or a bacterium or parasite, or in the case of cancer, cells that are no longer behaving as they are expected to inside a tissue. They're not listening to the signals that are going back and forth between cells in a particular tissue, the brain, for example. They are dividing when they're not supposed to. They are not dying when they've been asked to commit suicide in order to maintain the health of that organ. And there's chemical signals that are sent back and forth between cells constantly, and also chemical signals that reside on the surface of cells that help these cells here define whether a cell is behaving as it should in a healthy tissue. And I'll sort of highlight a couple of these cells. Dendritic cells, for example, or macrophages, are able to chew up a cell that's injured, infected, or behaving inappropriately. And its job is to present the proteins and so on that are inside that sick or irregular cell and present them to members of this set of cells. These are effector cells whose job it is to go do something about it. And so they take the identity that they've defined after chewing up these irregular cells and pass that on to these cells, and they'll generate, in the case of B cells, antibodies that will hunt down these sick or irregular cells or foreign invaders like viruses or bacterium and kill them. Or in the case of T cells, and specifically for cancer, CD8 positive T cells, will actually go door to door, so to speak, or cell to cell contact and kill these cells that are irregular or thought to be pathogens one by one. And so the whole thought of the immune therapy is to take this system, as complicated as it is, and direct it towards seeing cancer cells in such a way that antibodies and T cells can be derived to target those tumor cells and eliminate them. Now, this is not a new idea, as I've mentioned, and in fact, it's not a human. It's something that's been invented. The human body itself has been doing this since it was set up years ago. This immune system is constantly surveilling our body for tumor cells and eliminating them. And in fact, we've learned much about the communication between the tumor cells and the immune system, and realized that the tumors are actively trying to control the immune system themselves. They're trying to shut it off and to hide from the immune system, to hide from these CD8 T cells, to hide from dendritic cells and macrophages, and change the behavior of many of these cells so that the tumor can actually flourish in an environment where the immune system would normally be clearing. And so many of those breakthroughs in this understanding have given us the ability to now harness the immune response against the tumor. And here's just one example I'd like to mention about what people are doing. So this is an example that is maybe 10 or 15 years old, but is constantly being improved upon to the point where now clinically there's some really breakthrough responses being demonstrated in early experimental trials. Basically what happens is the patient would present in the clinic, and some blood would be taken, and the T cells from their blood would be removed. Those T cells would then be genetically manipulated using a number of different techniques, such that they would be rendered with the ability to see a particular protein that's on the surface of your tumor cell. So that way, this T cell now is programmed to go find and kill any cell that has that protein on the surface. Those cells are grown to a really high number and fused back into the patient in a process called adoptive transfer. And those T cells will then float through the body and repopulate that patient's immune response, but they're in such great number that they can find the tumors and start to cause those tumor cells to be lysed and propagate the immune response against them. So this has been done for B cells very effectively, tumors that arise from malignant B cells, either lymphomas or leukemias, and patients that would otherwise have failed all other therapies are actually walking out of the clinic with essentially no disease. They're essentially cured of their disease. So there are some, basically it shows just how powerful the immune system can be when it's harnessed appropriately. There are also some pitfalls with this strategy. One, you have to have an idea exactly what the tumor antigen will be, the means by which you target that tumor. You have to know that ahead of time because you need to engineer these T cells. And you need to be very careful with what you use to target those cells, because it can't be on any normal cells you'd not want to get rid of, because again, these are very potent cells, and if they find a cell that has that antigen, they'll kill it, and you could raise the possibility of an autoimmune response. So as long as you're very careful in defining this special tumor binding moiety, then the therapy works quite well. And the challenge moving forward will be to define that for not just B-cell lymphomas and leukemias, but other cancers. In fact, brain cancer is one that's being attempted now. So it's a very promising therapy, provided we can deal with some of those technical challenges. What I'd like to speak about is a little bit more of what we work on. Viruses called oncolytic viruses. It's a therapy, it's another biotherapeutic, similar to using the immune response, but it starts from the use of a programmable medicine, a virus that comes from nature that can be manipulated genetically so that it can specifically target your tumor cell and not your normal cell. So this is just an old schematic that defines the concept. Viruses are obligate parasites. They need a cell in order to be able to replicate. It has to steal the sort of machinery within inside a cell in order to make more copies of itself. If it can't infect into a cell and replicate its DNA or RNA and make its proteins, then the virus is dead in the water and gets just washed away in the organism. So there's a real battle between virus and host cell to figure out who's going to win. So the concept for the oncolytic virus is to engineer a virus such that it will only be able to win this battle inside a tumor cell and not in a normal cell. And I'll get into some of the ways in that is actually accomplished. Now, once the virus enters the cell, infects it, it causes more progeny to be made. So this is another key feature of an oncolytic virus is it's a self-propagating medicine, as opposed to a medicine that is delivered into the body at a prescribed dose and then begins to decay. This is a virus. This is a medicine where you deliver a virus, and at the place that you want it, at the tumor, it actually amplifies itself and can spread to, again, an adjacent tumor. And provided you set this up properly, adjacent normal cells will not be infected, and you propagate this chain reaction only through tumor cells. There are a number of different oncolytic platforms that are being tested based on different viruses and modifications to those viruses. They're in various stages of clinical development. Stage 3 is the final stage before it becomes clinically available on the market. Some are in very late stages. Adenovirus, vaccinia and herpes virus are all very late stage. This is a bit of an older slide. But there's some that are earlier stages that look quite promising and have properties better than the first generation, but they're making their way through early stage trials like VSV, measles, and poliovirus. So why are we excited about oncolytic viruses? Well, one of the things that is very impressive about oncolytic viruses is their tumor selectivity. They're acutely aware of the genetic differences between a healthy cell and a tumor cell. And they do that because they rely on the genetic alterations that are present only in tumor cells and not in normal cells for them to be able to win this battle, to be able to replicate and form their progeny. And those cell states, or those building blocks that the viruses need, are really only present in tumor cells. And so consequently, they are incredibly selective for tumor. So here's an example of a mouse tumor that's been set up. Again, we can see a tumor, but this now, the green is a virus that we can see. So the tumors themselves are red. They're set up in the brain, for example, and then the virus is injected intravenously, and it expresses a green protein. So when you look under the microscope, you can see the red tumors in the brain, and you can also see the green virus. And where it's red and green at the same place, where they co-localize, you can see sort of a yellow color. And so what we see here, in sort of a higher magnification, is that there's a boundary here between normal cells and tumor, this being the normal tissue, this being the tumor, and the virus can replicate right up to that boundary, but cannot cross that boundary because the conditions here are not conducive for the virus to replicate. Only those conditions are available in the tumor. And that's really critical, as I mentioned, this tumor selectivity in the brain, because we will significantly decrease the side effects of the therapy. Now, how is this set up? Now, it's beyond the scope, perhaps, of this discussion to talk about all the different ways that people have identified or engineered antiviruses to make them selective. But I put this slide here to show that there is not just one way, but there are many different ways, many different tools in the toolkit to program your virus to only replicate in a tumor versus not. And so, for example, there are mechanisms by which the virus requires signaling pathways, these sort of protein chain reaction signaling events that happen within the cell that are present in malignant cells, that are not present in normal cells. And as long as these pathways are on, you'll have a malignant cell perhaps, but you'll also allow the virus to replicate. If they're off, as they are in normal cells, then these viruses can try to get into those cells but not be able to continue their life cycle. And there's other ways where people are actually, rather than let the virus get in and then try to make a go of it and fail, they in fact retarget the virus to only be able to attach to your tumor cell and not your a normal cell. So the receptor for that virus will only be present on your tumor, and therefore the virus can only really attach and infect those cells. And there's quite a number of other ways, and these sort of little tactics and tools are continuing to evolve constantly, and people are mixing and matching them together to make viruses more selective for particular kinds of cancer, or safer or more potent. Now, another key component about oncolytic viruses is that they fulfill the need to target all types of cells within a brain cancer. So not just the drones, but in fact, many of these viruses, not all, but many of them, have been demonstrated to attack these cancer stem cells that have been derived from patients and grown in cultures. So this is something that's done in our group, where we can grow these glioma cells that have been harvested from a cancer patient, and we grow them in the way that Peter Dirks has defined some years ago. And these cancer stem cells we can then infect with one of our oncolytic viruses, in this case Farmington virus, that expresses a green protein, fluorescent protein, that allows us to track which cells the virus and how well the virus can affect those cells. So clearly it infects these cells incredibly well. And then if we wait a little bit longer, all those cells will be killed. They undergo apoptosis and die. So it's important that these viruses can not just kill the drones, but they can get at the root of these tumors as well. Another important thing is sort of summarized here in this reasonably complicated slide, and it's one of the fundamental principles we think underpins the efficacy that we see with oncolytic viruses, is that they're multimodal. They attack in not just one way at a time, as a certain drug might be designed to attack a certain the DNA, for example, of a tumor, and therefore make it difficult for the cell to divide. But if you're only attacking one way at a time, then the tumor cells will have a chance to evolve a way around that. As I mentioned, there's a lot of genetic heterogeneity, not just between patients, but within patients. And so we feel that the only way that you're actually going to effectively treat the entire tumor is to be attacking in many different ways at the same time. And that's in fact what happens with an oncolytic virus, just empirically. So for example, if this is a tumor in brown, and this is the healthy normal tissue, virus can be infused either directly into the brown tissue, into the tumor, or through the vasculature into the bloodstream. And we can see what generally happens in a tissue culture dish when we model this is that the virus will leave the bloodstream and attack the tumor tissue directly. It will replicate, and in replicating will start killing that tumor tissue. The other thing that happens is that it sets up what's called an inflammatory response. Some of these chemical signals to the immune system are released while this virus is starting to kill these cells. And that does a number of very key things. One, in John Bell's lab, a colleague here in Ottawa, his group has shown that what that does is it attracts a certain immune cell, this neutrophil, into the tumor area, and it chokes off the blood supply. It causes the constriction of these, or basically the plugging of the blood flow to the tumor, and it chokes off the tumor to its blood supply, and much of the tumor just dies off because it chokes to death. Another thing that happens is it recruits NK cells. And so if you recall in the slide where I mentioned a few of the constituent cells in the immune response, NK cells are attracted by these chemical signals from this inflammatory response, again, to leave the bloodstream and enter into the tumor, and NK cells will then recognize these tumor cells for what they are, cells that are abnormal. They don't have the correct surface markers and aren't behaving the way they're expected to in this tissue, this complex tissue, and they'll begin to kill those cells very rapidly. Another thing that happens, again, this is all happening at the same time there's graded response where virus enters the tumor and sets up this chain reaction of events. During the killing of these cells, these tumor cells, what you have is antigen, these proteins, gets released into the area, and it sort of drains down into an organelle that's involved in constantly sensing the proteins that are floating around in the body. And if some of these proteins are deemed to be foreign, proteins that are a little bit irregular, ones that haven't been seen in the body before, the immune system then decides maybe these are foreign and I should mount a response against them, and sends in the cavalry in the form of these T cells or B cells, and starts to target, as I've mentioned before, these tumor cells now based on these antigens, these proteins that were released. So it sets up a number of different events all happening concurrently at the same time, and that causes these catastrophic decreases in regressions of the tumor that we see, and I'll show you shortly. And it also causes long-term effects, because what I didn't mention before is that once the immune system, in the form of these T cells, has found a foreign invader, it has this ability to memorize the molecular pattern of that foreign invader, and it keeps that in sort of a library of memory cells. And if it sees that same invader again, it mounts a very, very robust response. Again, of these T cells, they just multiply like crazy and come and attack, so that you can't ever be sort of invaded by that same pathogen again. And it's the same is true for tumor cells. You then become immunized. You're immune to that tumor cell. And so consequently, we see these very long-term responses in our models, and we think that's also now happening in early clinical trials with these oncolytic viruses as well. So it's a robust attack, it's a multi-pronged attack, and we think that's why it's so exciting. And so this is just one example. I didn't want to put a lot of data into the slide, but this is one example of our work with brain cancer and some viruses that we've been working on for a half-decade now to optimize them for brain cancer. And this is a human xenograft model. It's a human brain glioblastoma tumor that's implanted in mice. And if they're left to be untreated, then these animals will all succumb quite quickly to the disease. And if they are treated either intravenously or intracranially with the virus, in this case Farmington virus, one of our lead candidates, we can actually cure some proportion, up to a third of these animals will actually be cured of their brain tumor. Now these are large brain tumors, and we have, this is one particular model, but we also model a tumor that's a little bit closer to the clinical setting in that it's highly invasive, and it's sort of summarized in all of the model work we've done. Essentially, it doesn't really matter what the model is, we can cure every animal benefit, but we can actually now start to cure tumors in these very difficult to treat cancers. So how does this compare with what's been shown for the two frontline therapies that are available now to patients? So if we benchmark it against what is available now to patients and look back on when these drugs were used in animal models, we can see that they are definitely beneficial, and that sort of phenocopies what the clinical experience has been. In both cases, they benefit patients, but in no cases do we see any cures. And so we feel now that we're actually able to have a medicine here that has a real shot at long-term, not just short-term, but long-term benefits to the patient. And we're now mounting an attempt at moving these to a clinical trial, first in man clinical trials. So I just wanted to have the last little bit to talk about what some of the early clinical trials are showing us with these oncolytic viruses. So is this something that's just a fancy of the lab, something that we can get to work against cells in a dish or tumors set up in mice, or are there real tangible evidence for these benefits being transferred to humans in the clinical setting? And so a colleague of ours, Matthias Gromeyer at Duke University, has been working with his particular oncolytic virus, and it's a poliovirus that's been modified genetically so that it's very safe to inject into the brain, but it's also very highly lytic against brain tumor cells. So he's found a particular way of generating this tumor selectivity. And he's now launched a trial that's about a year old, and this is an example of I was able to find that shows the results and shows that after a short time, this is a glioblastoma, after a short time you can see that there's a tumor in this patient, but several months later, long after the virus has left, in fact, we suspect, the tumor is continuing to shrink, and several of his patients have been completely cured in terms of radiographic output. So there's no longer, by MRI, any tumor that can be found at 11 or 12 months post-treatment. So it doesn't happen for all patients, but when it happens, it's quite remarkable. And it's a very long-lived response. And so we think that it's early days, and he's continuing to add patients to the trial, and is looking to try a phase 2 trial in a great number of patients shortly, and we're all really rooting for him, and we think that this virus looks very, very promising. There's others in the pipeline as well. Now that's just oncolytic viruses on their own. I'm going to leave you with one more concept that we are also very, very excited about, and it's a concept whereby we know that oncolytic viruses are engaging the immune response as part of their multimodal attack. And we also know that immunotherapy on its own, where we're manipulating T cells and vaccinating against tumors, has been also quite promising. Now, colleagues of ours at McMaster University, Brian Lichty, Yan-Hong Wan, and Jonathan Bramson, have actually decided that rather than wait for this engagement of the immune system that the oncolytic virus can launch, rather than wait for that to happen empirically or holistically as part of an oncolytic therapy, why not marry the benefits of oncolytic virus therapy with immunotherapy in a very direct sense? And they've done this by including inside our oncolytic virus backbones the protein, a tumor-associated protein, a protein that is known to be part of the tumor, right inside the oncolytic virus. So as a consequence of replicating, once the virus is infused into the patient, finds a tumor, replicates, it actually expresses the tumor-associated antigen as a vaccine. And this way, when the virus is infused in the patient, it not only chews up the tumor and launches that multimodal response I spoke about, but it also expresses these antigens and shows them to the immune response in the lymph nodes and the spleen and so on, and causes the body to be vaccinated against that tumor antigen. And in this way, you get sort of the marriage of both of these things. And I'll just leave you with sort of one example of the benefits of this. So in this case, what they've done is use a tumor model that is in fact resistant to the oncolytic virus. The virus can't really infect this particular tumor very well. And they set up this tumor in the brain, and they treat with an oncolytic virus, and it's not shown here, but basically it doesn't cause any benefit to this tumor model. So the animals will succumb. If they vaccinate, if they use an immunotherapy against this particular tumor antigen, they vaccinate those animals, they can see most of those animals benefit, and some do really quite well, on the orders of what you would see with temozolomide that I showed you on those previous figures. And you can see that if you measure the number of T cells now that are specific for your tumor, you can go from nothing to detectable. But if you use this concept that they've devised of an oncolytic vaccine, where you put in this tumor antigen within the oncolytic virus now, and use these in combination, you vaccinate and then you put the oncolytic virus in, you generate really high responses to the point where a third of every T cell in that, this is in a mouse, is now targeted against your tumor. Well, now you see that even in a situation where the oncolytic virus can't do any of the work, the vaccination now becomes very effective to the point where we're starting to actually cure patients just with the immune response, because it's driven by this oncolytic presentation of the tumor antigen. So this essentially is bolted on to the efficacy you see with an oncolytic, and then that's how we think we're going to start to build really truly clinically effective medicines, by adding one modality, one effective sort of capability on top of existing capabilities, till we get to something that's truly effective for not just a single patient but many patients that present with this disease. So so I just wanted to sort of summarize what we talked about today. Yes, brain cancer therapy is challenging, and it's challenging for a number of reasons that we've gone over. And the clinical toolbox of these therapies, though, is not iOS. That's some sort of subliminal Macintosh message. But the clinical toolbox that is starting to be available at the experimental level, and hopefully soon at the clinical level, is also expanding rapidly to meet these challenges. As we understand how the disease functions, what are the key components of the disease, we're involved in how the virus, the cells spread through the brain. As we understand more about how the body responds to it, the immune response, how we can manipulate the immune response, and then also as we know more about how viruses work, we're now putting all these pieces of information together to hopefully, one by one, meet all of the challenging criteria that brain cancer present with more and more sophisticated therapies. As I've mentioned here, and didn't really go into enough detail yet for time reasons, early results look very, very promising. But of course, much more can be done, and that's what we're hoping to do with funders like the Brain Tumor Foundation of Canada, as well as these other funders. They're funders of our work here in Ottawa and throughout the country. And there's also more information that's available through some of these outlets we have from my group and other collaborators group throughout Canada. I encourage anybody who's interested to contact us or other collaborators in your local area to find out more about what's being done at the bench level, at the basic science level, to try and meet these clinical challenges. And with that, I'll take any questions. Thank you.
So great, Dr. Stodel. Thank you so much for this great presentation. I know for myself, not being a physician or a researcher, you really presented the information in a way that is very easily understood. We do have one question and one comment. The question is, can this virus also do its work when tumors are in remission, or must one wait for it to become active again?
Oh, that's an excellent question. And what I suspect is that when one is truly in remission, where there's absolutely no cancer to be found, then that's not the time where this virus should be injected into the patient. Because as I mentioned, these viruses need tumor cells to be able to grow and to be able to launch all of these multimodal responses, the immune responses that we've talked about, the effects on the vasculature and so on. So if there's no tumor for the virus to replicate in, then you're not going to see these benefits. And so we envisage that, in fact, one day these therapies will be frontline, and so that people that have bulky tumors will receive these therapeutics and launch all the things we've spoken about. Now, having said that, it's also true that while people can be in remission, that doesn't mean they're completely disease-free. In fact, there perhaps might be residual tumor in those people. In fact, that's why most people in brain cancers will reoccur, because there is residual disease. And so we think in the case of brain cancer that, in fact, the first trials that we'll attempt to do will be in patients that have reoccurred or are between, have received their surgery and are waiting to, essentially are in remission, but waiting to find out what happens next. So I think it's an important point. These viruses need tumors for their life cycle, and so it would be pointless to put them in where there was no tumor. But unfortunately, in many cases, there is tumor material there.
Great. Thank you, Dr. Stodel. There's a couple other questions just in follow-up to that. If there is still tumor there, but it's not growing, is that a good time? And how does somebody get into one of these trials when the time comes?
Right. Yeah, no, I think that would be a great time. In fact, you know, one of the things that we are really excited about is because we know the immune system is so important, it's important to have a healthy patient, one that still has a robust immune response that they can bring into the fight. So when patients are sort of very far along and their immune system is weak, we think the oncolytic will still have its benefits, but we won't really maybe able to tap into the extra benefits of having a strong immune response that will help also target our tumor. So if there is a slow-growing tumor, then that means the patient has time to mount an immune response. I should also mention, too, that immune responses take some time to mount. They take months to mount. So slower-growing tumors are a perfect situation in which the virus can have a place to replicate, launch these responses. You can regress the tumor, but also give the immune system enough time to mount an effective response and be able to get rid of residual disease. So I think that's sort of the way we see it. In terms of how to get on trial, well, I think the way to do that is to contact research centers to find out what trials are available, and also, of course, speak to your physician and ask them about what trials are available that they know of. You could also bring bring some trials to their attention, find out if they know about them or can help you get introduced to those trials at those centers. But your primary care physician is the best place to start.
Great, and absolutely right, Dr. Stodel. And we also have a section on our website where there's a various web links to clinical trials in Canada and in North America, so I could always send that to those of you who are interested to find that on our website. We have a few more scientific questions. I'll give you at least one of them right now, Dr. Strodel, and if we don't have time to go through the other ones, I will connect you with this gentleman via email. But one of his questions is, why haven't the CSCs been grown as spheres? Looks like cells have just been grown in bulk, and you won't know if you truly targeted the CSCs unless you sort for CD133 or CD15.
Right. So I assume you're referring to the pictures on our slides. And so these are CD133 positive cells. They've grown. So Dr. Dirks' group some time ago has modified the way cells are grown rather than neurospheres. They grow them in laminin, beds of laminin, allow the cells to actually grow flat. It doesn't mean that they're not stem cells anymore. It's just they're grown in a slightly different way, and they present with all the same markers that you'd expect in a cancer stem cell. In fact, they maintain those markers better than they do in the spheroid form, and especially after they've been transplanted back into an animal. They are more robust in generating tumors, so they seem to be more potent. So this is sort of the new way that those cancer stem cells are grown, and we're sort of following along and using the great work that his group have done to define these conditions.
Great, thank you. And another question was, has anyone done secondary injections with tumors to show that the oncolytic viruses truly can serve as a vaccine?
Absolutely. In fact, that's what that's a good point. I didn't want to get too technical about it, but the immune response increases greatly the more times it faces that particular invader. So when you've got this sort of memory response against a particular invader or a malignant cell, if you show them that identity, that genetic identity, again, the immune response gets even whipped up to a higher sort of level. And so we definitely think that the best way to do this is sort of in a prime boost fashion. It's sort of what I showed in the last slide, where you vaccinate and then come in with an oncolytic virus which vaccinates over and above that, sort of boost the response, which is a sort of a classic way that we generate responses against foreign antigens. You know, as a kid, you go in, you get your vaccine against a virus, and then you get the booster vaccine. Well, we think that's exactly the way it's going to go in cancer, and that's why we're also really interested in making sure we build out a whole panel, a stable of these oncolytic viruses, so that we can continue to give patients different viruses each time that are focusing the immune response against the tumor and not against the virus. And so we can give continued care as opposed to sort of just one shot of the patient.
Perfect, thank you. And just one last question, Dr. Strodel. How does this work with the brain, as microglia are the resident immune cells in the brain?
Right, and so microglia, no question, will pass antigen to the rest of the immune system to help launch immune responses. But there's this, I don't want to get very technical, I suppose, but there's this concept where the immune system in the brain is sort of detached from the immune system in the rest of the body, and I think that is an idea that's sort of eroding. There is an immune privilege in the brain where the immune system is kept quite dampened because, again, it's a delicate, complex tissue. You don't want the immune system causing any unnecessary damage, so the threshold to launch a response is higher in the brain, but the brain is perfectly capable of launching very, very robust immune responses. And there are a number of cells from the rest of the body, dendritic cells, B cells, T cells, NK cells, neutrophils, can rush into the brain if necessary, if it's deemed to be necessary to deal with the insult. And that's all done through chemical signals, so-called chemokines and cytokines, that help recruit the army necessary to deal with the challenge that the body is sensing.
Great, thank you so much. Such great questions and great answers as well. And just one last comment from somebody who was signed on to the webinar today, Dr. Stodel, and she wanted to let you know that as a brain cancer patient, I wish to commend and thank you and your team for doing this research work. It is exciting and encouraging to see that there are several concepts being devised and investigated to improve longevity and quality of life and potentially even a cure for some. The webinar was very informative for me as a patient, and I thank you. And we thank you because this presentation obviously has provided lots of great questions and a lot of great information. And as I noted at the beginning of the webinar, this presentation has been recorded and it will be up on our website within the next few days. I will send an email out to everybody who did register to let them know that it is up on our website and ready to view at any time for free. And just a reminder that our next webinar is with Dr. Brenda Sabo from Halifax. And her presentation is going to be entering into difficult conversations with patients and families, avoidance is not a strategy. Now, this particular webinar, the intended audience is for healthcare professionals. However, patients and families are more than welcome to sign in to the webinar. And we also want to encourage everybody to visit our website, the Spring Sprint section of our website. Dates are now posted on each city's page, so you can save the date for the closest event to you. So once again, Dr. Stodel, thank you so much for your time and for participating in our webinar series. And we look forward to collaborating with you again in the future.
Well, thank you very much. That was a great treat, and I welcome the opportunity.
Great. Thank you. Take care. Goodbye, everybody.
Bye-bye.